It's been an exciting 3 years for the Foundation. We lured Kathryn Davidson and her valuable IPS skills and expertise back to Australia from America. The movie Stem Cell Revolutions toured Australia with screenings and panel sessions for schools. Community information events are helping patients sort the hype from the hope of cell therapies for conditions such as vision loss, multiple sclerosis, type 1 diabetes, and the list goes on. We also last year brought out international stem cell pioneers Irv Weissman and Anne Tsukamoto to visit Australia to meet local scientists and to present a series of public forums. We've worked with Stem Cells Australia to produce an up-to-date information handbook for patients considering their treatment options. The magazine-style Stories of Australian Stem Cell Science is celebrating local research success stories, and last year, of course, we awarded the inaugural Metcalfe Prizes for Stem Cell Research.
My name's Christine Wells and I work at the University of Queensland in Brisbane, and I also work at the University of Glasgow in Scotland.
My name is Jose Polo. I'm a group leader and associate professor in Monash University at the Australian Regenerative Medicine Institute and the Department of Developmental and Molecular Biology.
My name is Kathryn Davidson, and I'm a stem cell researcher at the Centre for Eye Research Australia, and I'm interested in understanding how cells are affected in macular degeneration, where cells in the back of the eye die, and it's not understood why these cells die.
My research is really about bringing together different types of stem cell data in different types of formats and compiling it in a way that's easy for clinicians and other stem cell researchers and the general public to be able to look at in a simple and intuitive way.
My research concerns what is called the epigenome. You can imagine these as small chemical signposts that are tagged onto the genome. The epigenome is critically important in controlling what a cell does in stem cells that are derived from and produced from specialised cells.
To be honest, I like the way that the research is performed in Australia.
Australia is a great place for stem cell research. There's a great community of stem cell scientists in Australia who genuinely work together in a real collaborative environment. And I think Australia has a strong history early on in the human stem cell field.
How cells define their identity, and how a cell becomes the type of cell that it is, and how we can manipulate that.
My research is really about bringing together different types of stem cell data in different types of formats and compiling it in a way that's easy for clinicians and other stem cell researchers and the general public to be able to look at in a simple and intuitive way. An analogy I've used recently is if I asked you to describe Melbourne and you described your local suburb and it was St Kilda, I'd get a very different description to someone who was describing the centre of Lygon Street. So we might call something the same thing, but what we're actually meaning are two quite different experiences. So to be able to bring those pieces of data together and compare them, we can really ask, how similar are they? What features do they have in common? Or how different are they really?
We were looking at cells that had originally been, say, skin cells, and they had been prompted or pushed to turn back to a stem cell-like state. What we found is that there were hundreds of places in the genome where the induced pluripotent stem cells had this pattern of tags, epigenome patterns, that looked like the original skin cell that they came from. So in a way, this was a memory of the cell that they originally came from, and what we think is this may carry with it some of the functions and particular characteristics of that specialised original cell. So we'd love to be able to try to erase this memory, to make these stem cells forget what they once were. So we're working on these tools for what is called epigenome editing, and we think this might be a really new way to erase the memory that's present in these adult stem cells.
So by enabling data to come together, we're really providing an engine of discovery. You can often see patterns in one type of stem cell or one type of stem cell data, but actually it can be very hard to know how similar two things which are called the same thing but might actually be quite different.
So stem cells are going to be critical to developing some of these new therapies, and I'm very excited about some new opportunities to work with leading stem cell experts in Australia to help develop more translational therapeutic products.
Stem cell research is really blue sky research. It's an incredibly exciting area to work in. Government grants tend to be really directed at a particular outcome. So we need a broad funding base. We need interested philanthropists, and we need businesses to come to the table and help us explore some of these bigger questions, because I think these are the areas that are going to open up really dramatic discoveries in the near future.
Come and meet with us so we can explain you what we can do and why we do this.
There have been a number of funding cuts recently in Australia from the federal government, so there's a huge need to take these scientists who've been mentored and trained and invested in for their education and support the next generation of scientists coming up through the ranks.
I don't ask you to put your funds in places where I will not do it. I changed my career. I was a cancer biologist that was doing very well, and I truly believe that this was the field to be if we really wanted to make a change.
We clearly face challenges with health and maintaining good health, and funding and support of stem cell research is really critical because it's going to open up a whole new toolbox of abilities in understanding not only how our bodies work, but in repairing them and fixing them.